My name is Carol Gabrielli. I'm privileged to facilitate this session where we get to hear from more Exito colleagues who are further down the path, and to learn from them, and to also have opportunity to ask questions. Many years ago, I learned, as a result of some experiences that I had, that in some languages, there is no word for work, that we simply tell stories about how we live our lives, and out of that, persons get to know what's important to us and how we spend our time. And I think today's panelists will exemplify that, of what they find keenly interesting and how their passion follows their energy, follows their attention, follows their daily lives. So we have 5 minutes of presentation with each of them, knowing that at the end we'll have opportunity to ask questions. The panelists will go in the order that is printed in your agenda. Sean, one of his many talents there in the back, will be to help keep time for the panelists. He has some cards there that I think offer 3, 2, 1. Sean, is that correct?
3, 1, 30 seconds.
3, 1, 30 seconds. Okay, that's something to remember, panelists. 3, 1, 30 seconds. All right, so we ask you to bring your attention to the panelists, certainly to have your notebooks, your lovely notebooks ready so that you can maybe jot down questions as they surface for you. And or just comments in their own storytelling. There may be some valuable phrases or words or just images that they use that you want to remember. And so, a warm welcome to our panelists. Betty, thank you for being first and your willingness to go. Thank you.
Oh, this is a little high. Good morning. I am thrilled to be here. Such an energetic group. Okay. Right? I think it's an energetic group. So my name is Betty Izumi, and I am in the School of Community Health, though the School of Community Health and OHSU just formed a School of Public Health, so I'm now a faculty member in the School of Public Health. So just by way of background, I did my undergraduate degree at the University of British Columbia. I'm Canadian, and I just received my U.S. citizenship last summer, so I get to vote this year. I did my master's degree in public health at UC Berkeley, and I also became a registered dietitian through the Berkeley program. I worked for 4 years in between there, gaining some work experience, and then went to Michigan State University and did a PhD that was very interdisciplinary in nature. And then I went to the University of Michigan to do a postdoctoral training on how to use a community-based participatory approach in my research. So that's what you'll see reflected in my slides today. Oops. Hey, now technology is not my strength. Hold on. Oh, okay. Is it on? Oh, there we go. There we go. Okay, so my approach is community-based participatory research. So what this means is that in all of my work, I work with the community to identify the research problem, and they're involved in every single stage of the research process, from data collection all the way through dissemination. The primary themes of my work are health inequity, so identifying and working with communities that are very vulnerable and trying to eliminate health inequities. I'm a registered dietitian, so I focus on diet quality. I'm interested in the food environment and how the childcare food environment or the neighborhood food environment impacts diet quality. I'm interested in sustainable food systems, so this means developing interventions that also can promote a more robust and resilient local food system. And then I also work to prevent childhood obesity in my research. So I'm just going to tell you about 2 of my projects. The first one is CSA Partnerships for Health, and a CSA is community-supported agriculture. So many of you may be members of a CSA, but you pay upfront in the spring about $500 or $600 to a farmer, and every week you get to pick up a share of vegetables, whatever they grew whatever they're growing at that time. So in this project, I work with various farms in the Portland metro area, and each farm is paired with a federally qualified health center. And so that's a health center that serves very low-income populations. So the members of the CSA program, and we just kicked off the data collection, they pay $5 a week for $20 worth of vegetables. And the rest of the $15, the other $15 is actually subsidized by grants. But the vision of our project is that one day, health insurance companies might be willing to subsidize the cost of eating vegetables through a CSA program. So this is another project. It's called Snacks, and this is a project in Eastern Oregon. So some of you may be familiar with Eastern Oregon, but I work in Union County, which is right next to Idaho. And this is a project that is in collaboration with OHSU faculty. And in this project, we engage middle school youth to create healthy snacking zones in and around their schools. So in these communities, there's one school and one store, and over the last 4 years, we've been working very actively with youth to change the food environment both within the school and then also improve the healthy food choices that are available in their local food stores. So you can see here that in this particular school, their mascot is a panther, and the kids actually came up with the idea to create a panther snack section at their local food store. And these are the kinds of snacks that are now available. And when we started the program 4 years ago, there was really virtually no healthy snacks available or healthy beverages. So we feel really proud of the students and all the work that they've done to get healthy snacks and beverages in their stores. So I was asked to address how I support students. So if you're working with me on a project, and I've worked with an Exito student in the past, and I work with many graduate students, but I really try to help students gain professional skills and to network with researchers and community partners and just create a really big network, because that's what you really need to be successful in the world. To gain qualitative and quantitative research skills, to develop written and oral communication skills, and then also students gain opportunities to participate in manuscript development and presentation of research findings. And then in terms of becoming a successful student researcher, I combine this with what my expectations are of student researchers. So I expect students to, and these are the things I think you need to be successful. So to take initiative, if you don't know the answer, then just go find out. Anticipate your research team's needs, be a team player, do your best work all the time, and be flexible and go with the flow, because in research, especially community-based research, things change all the time, and there's much that is unpredictable. Ask questions, and then finally, so I have a daughter who is turning 10, and for the last 2 years, every single day before I go to work, she shakes my hand and she says this: work hard, work smart, and work consistently. Thank you.
So I am Sonnet Jonker. I work at OHSU. I'm in the cardiology department. And I list here some of the other investigators in our research learning community, because a lot of times I'm asked if I'm an MD or if I'm a doctor. I'm not an MD, I'm a PhD. So the senior scientists in our group, okay, pointer's not working, but the senior scientists in our group are a mix of PhDs and MDs. George Giroux is a cardiologist, Lowell Davis is an OBGYN. We have a group of mostly PhDs, investigators, and a surgeon. Fred DeBian is a surgeon. We have postdoctoral fellows and undergraduate scholars and technicians in the lab as well. So I study heart development. It turns out that the cells in our hearts that do the pumping work don't divide or proliferate anymore in our hearts. And this is the reason that heart failure and heart attacks are so bad, because when those cells die, the replacement is scar tissue instead of working cardiac myocyte. So if you are an individual that has a lot of cardiac myocytes in your heart and you have some disease, then it can be not so bad because you've got a lot of cells, and that when some of them die, they can just grow a little bit bigger. But if you only have a few cells in your heart, few cardiac myocytes, that when they die, the remaining cells can't adequately compensate for it. So because the only time that these cells proliferate and make more of themselves is in the fetal period, I study that period and trying to figure out what causes the proliferation and what causes it to stop. So this just sort of illustrates the life cycle of cardiac myocytes. In the fetus, the cells are very proliferative, but it's hard if we know that a baby has congenital heart disease, it's hard to treat the fetus. People are doing fetal surgeries, but it's really risky for the mom and for the baby. So it would be really great if we could do something to help the baby after it's born. And so one of the things that we're trying to figure out is the transition period between proliferative and non-proliferative cardiac myocytes. And that happens just about the time right before the baby's born. So if we can extend that period a little bit longer, then we'd be able to give babies treatments instead of trying to do it for fetuses. By the time you're an adult, it's pretty much too late. There are some stem cell therapies, but there's been a lot of hype about them, and it hasn't actually worked out very well. So this is just a diagram of how cardiac myocytes grow and mature. So at the top, we have a cardiac myocyte with the normal number of chromosomes. That's called 2N. And it can duplicate its DNA, just like any cell that's proliferating, and then undergo cytokinesis, and you end up with two more 2N or mononucleated cardiac myocytes. And this is the cycle that happens in the fetus. The cells proliferate and make more of themselves. But as birth approaches, the cardiac myocytes duplicate their DNA, but then they don't undergo cytokinesis. So then you've got one cell with two nuclei, which is pretty cool. It's really easy to see these cells. You just look at them and there's two nuclei in them. These cells start to mature and they retain this phenotype with multiple DNA copies into the adult. So one of the things that we're looking at is all of the other maturational changes that are happening at the same time as this end to proliferation. So one of the things you might not know about is in cancers, the cancer cells like to undergo glycolysis. That is, they like to consume carbohydrates. And it turns out this might be true in fetal cardiac myocytes too. So over on that far side, you can see the top is all the fetal characteristics, so it likes to eat sugars. But as the cells mature, they prefer to eat fats. So in our hearts, the cardiac myocytes very much prefer to eat fat over sugar. Some other maturational changes that happen around the time of this change in proliferation is illustrated there. It's the cells actually undergo changes in what sort of genes are being expressed and how they function. So they like to use more calcium from inside the cell, for example, instead of outside the cell. So we can look at how those changes might affect the proliferation. And then there's also a period of what's called apoptosis. The cells, some of the cells die off about the time the baby's born. So if we could protect the hearts by preventing that die-off, then we might be able to sort of increase the strength of fetal hearts. So just real quickly, some of the techniques that if you were working in my lab, you would be culturing fetal cardiac myocytes from sheep and extracting their RNA or proteins. That just shows quantitative PCR and comparing different treatments. So that's the end of my time, but these are just some of the people in my lab doing lab things, so doing science. Thank you.
So I'm Marilyn Mackiewicz. I'm a chemistry professor in the department here at Portland State University. Just a little bit background about myself. I was born and raised in the Caribbean. I lived there for 16 years, and then I moved to the United States in New York with my parents. I went to Hunter College, where I got my bachelor's degree in chemistry and psychology, and really liked chemistry a lot, so I pursued that for my PhD at Texas A&M University. I moved to Portland a couple of years ago. It's been almost 10 years, though. It's not a couple. With my husband, who's also a chemist. And I started my postdoctoral research at Portland State and then branched out into my own research area. So in my lab, we design nanostructured materials for biomedical applications, and that's a really generic way of encompassing all the different things that we do in my group. I can't do any of the work that is involved in my group without my students, and just shown here is a couple of my students that are in my lab currently. My lab is really unique in that there's no postdoctoral associates or graduate students. Everybody in my lab is an undergraduate student that I recruit out of freshman level chemistry. So it's unlike any lab that you'll see at PSU. And I'm really, really fortunate to have these students, and these are the students that drive graduate level research projects in my group. So I would say that's one of the most unique things, is that they don't work on undergraduate level projects. They are my graduate students, and they're undergraduates. So I kind of highlighted 4 major research themes in my group. We work on developing nanomaterials for drug delivery and imaging of cancer. We also work on a project with the KCI Institute in macular degeneration. And we have another project that is part nanoparticle based, but the other part is developing assays for Alzheimer's disease. And for all the work that we do in nanomaterials, we look at their safety and their toxicity and their environmental impact. So with the cancer project, we're looking at building these nanostructured materials that would allow us to have multifunctional capabilities, meaning that we can use them as imaging agents as well as drug delivery platforms to enhance the uptake of drugs to kill cancer cells and image the therapeutic effect. And this is a partnership with my collaborators at Oregon State University, where they're developing the therapeutics, and OHSU, where we're looking at nanomaterial uptake in cancer cells. With the KCI Institute and the Oregon Primate Center, what we're looking at doing with them is building imaging agents where we can tag regenerative retinal cells that repair macular degeneration, because we want to understand how those cells repair the process, and we'd like to be able to image that. So at the KCI Institute, they develop the imaging modality or the imaging instrument. We develop the imaging materials in my lab, and then we work with the Oregon Primate Center to image those labeled cells in the eye. So that's a fairly recent collaboration within the last year, and we're hoping to submit an resubmit an NIH grant that would allow us to push the technology further. So for our drug screening platform, we're developing for Alzheimer's disease. We'd like to be able to develop a simple assay that we can screen hundreds and thousands of drugs that already exist to repurpose them for Alzheimer's disease, because this is a challenge. So we work with another group of people at OHSU. My collaborator there is Philip Copenhaver, and so we can build our biochemical assay, develop some hits, and they can test it in cells and tissue. And we're also looking at trying to understand how the whole process of neurodegeneration takes place. So we're doing some mechanistic studies as well. And all of the materials that we build, we want them to be safe and biocompatible with minimal environmental impact. So we look at how these nanomaterials are built, and then we study them, their in vivo toxicity. Wow, 5 minutes goes by really fast. So you can ask me all kinds of questions about mentorship and sponsorship. I care about 2 things, well, besides my family, maybe 3 things. The first is I really love research, and I like sharing that experiences, and that's why you see my lab is composed of undergraduate students. mainly. And I really care about mentorship. So everything that we build is designed to train students to do research and develop themselves professionally so that when you leave your undergraduate level experience, you can go on and be very successful in your career. So these are a whole bunch of things we do, and I'm happy to provide those. Lab expectations, do hard work, listen to your mentors, and collaborate with everybody and be safe, because we're in a chemistry lab. And I think that I have like 1 second, that's it? No? I'm cut off. So you can ask me all about mentorship more specifically if you want during the discussion, or come up afterwards. Thank you.
Good morning, everybody. I'm a social psychologist, and my research has to do with social psychological aspects of health situations. So social psychology is about basically how people's thoughts and feelings and actions influence other people's thoughts and feelings and actions. In other words, it's everything, right? But I'm particularly interested in healthcare situations, and there are a few reasons for that. One is that I'm interested in discrimination and how people are experiencing circumstances in which they think they may be treated unfairly, and turns out this happens quite a bit in the healthcare system. The healthcare system, I think, is important in part because it's a major social institution. It's a major part of American life. And when we're sick, we're vulnerable. So we aren't necessarily at our strongest to be able to attend to how well we're being treated when we're also concerned about, the physical health concern that we've got. When we don't get the optimal care to which we are entitled as people, then this has proliferating effects. In other words, we might feel unsure about whether to follow the protocols that are recommended to us. We might be unsure whether to stay with the same provider or switch providers. All of this ends up affecting more things that affect more things that affect more things, right? And gets in the way of our wellness. So my research interests are on people's experiences of discrimination and healthcare, and I'm interested from the angle of looking at discrimination as a stressor. And so I'm interested in using social stress models to understand people's appraisal of their situations, and to understand this in the context of people's ongoing lives, their ongoing relationships, and their social roles. I'm particularly interested in what we call ambiguous microstressors, or you might be familiar with the idea of microaggressions. So a lot of times we can't tell whether something that just happened was a little off or not, and not being able to tell, in the way I'm thinking of it, not being able to tell whether the way we talk about it on our team is, was that a thing? When you can't tell, was that a thing or not, that kind of becomes stressful in itself. And so the ambiguity of a situation becomes kind of its own topic. So the research that I'm moving forward on is looking at literature to try to understand the ongoing pressure that people might feel when they feel they need to be ready for these ambiguities in life, and also how we appraise or how we make sense, was that a thing or not, particularly in a social context. So if I do a data collection this coming year, it would be in the spring, or it might actually be next year after I get a chance to work with some undergrads on a context of thinking about these different ideas in the literature. So this coming year, I've probably got a couple of different things going on. I've got some Wednesday afternoon database work where we're working on developing the literature bibliographic databases. But then I'm also going to have a Tuesday night journal club when we pick a journal article, we read it by the next week, or 2 weeks if we do it on a 2-week cycle, and we come together then and we talk about
what the research was, what was their understanding of stress, how did they think about ambiguity, what kinds of microstressors or microaggressions did they look at, what did we think of the research that they conducted, how does this match against our own lives, what would we have done differently if we were the researchers, that kind of thing. So I'm thinking that will happen over the next, starting in September, late September, over the next many months. So my support to scholars would be support in the short term, support in thinking about research questions and eventually working toward a data collection. Probably my next data collection would be looking at microaggressions in a campus context before I go to a healthcare community setting. And I was invited to give advice, and so I tried to hone it down to one thing, and I think my advice is to avoid embarrassing your research supervisor. And it's like, oh great, how do I do that? So let me put it positively. Showing public respect for your research supervisor is something that research supervisors really love. So, so when you're talking to the office staff about your doctor, your supervisor, your research supervisor, refer to them as Dr. So-and-so or Professor so-and-so. That's a helpful thing, So that's my suggestion. Thank you very much.
Wonderful, thank you. Christina, welcome. Thank you. And then questions, so stay tuned. Christina, would you like this? Okay.
Okay, perfect. All right. Hi, everyone. It's so great to be here. When I was asked to come and share a little bit about my research, it got me thinking back to my first experiences as an undergraduate research assistant. And so I actually started working by being a research assistant in 2 different psychology labs, one that was focused on clinical psychology and improving the mental health of Asian Americans, and another one that was really quantitative focused and working with big data to try to look at new statistical methods. And so when I just think about the space that BUILD EXITO has created, I think it's just such a wonderful environment, and I hope that you get so much out of it. And so just a little bit background about myself. My undergraduate was in psychology at UC Davis. I got my master's in clinical psychology because I was so sure that I wanted to do therapy and teach and do research, and figured out that working with folks one-on-one, I was not going to save everyone as fast as I wanted to. And one of my advisors actually suggested I turn to public health, and that's what I did my doctoral work in. And then, similar to Betty, I completed a postdoc where I really wanted to learn more about community-based participatory research and learn how to use those skills throughout. So my research is really focused on sexual and reproductive health disparities. I'm interested in understanding why these disparities exist, and then really trying to figure out what can we do with them through intervention research. And so when we think about sexual health and reproductive health, we see lots of health disparities, particularly among LGBTQ populations, Latino communities, and Black communities, and these are the groups I tend to partner with and work really closely with. I also, too, appreciate Betty's example of what community-based participatory research is, and that's the same approach I work with, too. So working with these communities, identifying priorities, and working with them through the entire research project is just a really important piece for me and how I conduct my research. In terms of my interventions, this is how we figure out ways in which we can try to improve folks' health and how we can try to address these health disparities. And I generally think about things in 2 forms, either the role of technology or the role of social networks. And so coming up here and seeing the issues with the clickers, I was like, oh golly, how embarrassing will it be for the technology researcher to have a technology issue? So I'm also very appreciative that all got worked out. And so I'll just give you 2 examples of projects that I've worked on. So there are social and sexual networking apps available for a variety of communities. One of them I worked on is called Grindr. You could think of this as Tinder for folks, for men who are interested in meeting other men for a variety of reasons. And so we looked at how we could encourage HIV testing through these spaces. And what we found that people were not only willing to engage this way, but it also led to some really positive results. The other example I'll give is around social networks. And so for this, we think about a lot of how everyone is connected to someone else. And so what can we learn and how can we use these social networks to improve health or encourage community? So similarly tying into Kerth's work and thinking about these spaces. And so for one of these projects, I was working with a group of Latino men who were part of soccer teams. So again, social networks. And we trained key folks there to be lay health advisors, or someone who would be there to advocate for the health, serve as a resource, and provide referrals to a variety of health or social conditions folks were working on. So those are just two examples of the intervention work that I've worked on. I was also asked to talk a little bit about how I would support students. And so I put up this figure because this would be my ideal imagery of what I'd like to do, where the figure in white would represent you as a student surrounded by all of this different types of support. And so that support might be peers that are working alongside with you, students who are at the same level, or perhaps educationally further along, and other research faculty and other faculty here, both at PSU and at other universities. I would personally want to support you by encouraging you to a lot of things: people, ideas, concepts, opportunities, and just trying to provide a really open, friendly, and inviting space. I was also asked to talk briefly about expectations, and so professionalism, I think, is just a really big one. Show up on time, show up ready, do what you say you'll do. Also, similarly to some of the advice that I was given before, be inquisitive. When you have a question, ask me, ask others in the lab, ask Google. Try to figure it out and get that answer. Take that drive. Try your best. And then finally, honesty. If something isn't working out, whether that's I'm having a hard time meeting this time commitment or there's other things going on in my personal life, let me know, let others know, so we can problem solve and work this out and figure out ways that we can make this the best situation possible. And then finally, guidance. I'll just highlight the bottom two. The first is advocate for yourself. And so I'm so excited that you guys are going to be doing elevator talks. I think those are so critical in knowing what is it that I want, and so you can keep that in your mind, and what am I interested in, so you can bring that forth and just be ready to talk about it and push that piece forward. And finally, being open to feedback. I think everyone in this space is really excited to be here to work with you. And whenever you get feedback, whether it's something that you might not have wanted to hear or something you wanted to hear, really knowing that it's coming from a place of kindness and just wanting to support folks and help you develop to the best that you can be. So just being really open to that and those learning opportunities. And I think I'm out of time, so I will end there.
Wonderful panelists, thank you. Jen, do you need me? I don't.
Oh, yes.
Great. Okay. For 4 or 5 seconds, what I'm going to invite us to do, in whatever way you want to interpret this, is to just wiggle. Just wiggle your body, wiggle your head, wiggle your hands. Just get your blood moving in a new way. There you go. Good. Look at the current scholars back there modeling for us. Look at that. All right. See, that's what 1 year in Exito offers. You can videotape, you can keep time, and you can wiggle. That's what we're going to put that on the brochure for next year. Fantastic. All right. Well, what we'd like to do in our efforts to make sure that we're hearing all the good responses and the good questions is to utilize the microphones among you, and then, of course, to utilize the microphones among the panelists. So I'm not going to say, do you have any questions? I'm going to say, what questions do you have? Because no doubt, given the bounty of information and passion that we've heard, much is bubbling out there. So we'd like to see what has been percolating for you. So what questions do you have? Who'd be brave enough to be our first question asker or inquirer? Thank you.
So for those of us who are distantly challenged, who aren't from this area, how do you think, if we're interested in your subject that you are researching and such, how do you think we can come in contact with you or be more involved in your project?
Thank you, Laura.
So at least for my part, I really love to write. And so I actually write a lot of emails to even the current people in my lab. I'll go talk to them, and then I'll go and sort of think about it some more and maybe summarize what I said and extend on it and give references in emails. So that's something that I like to do. And if a student was interested in my lab and wanted to correspond with me and maybe sort of do a little journal club or something like that by email, that would be something I'd be happy to do.
Yeah, I think that with community-based work, it's a little bit more challenging because I really believe that FaceTime is so important. But if you are interested in working at least on some of my community-based participatory research projects, I'd be happy to figure out a way to loop you in. And when you're in town for events like this, those are really great opportunities to go out and do field visits and meet with community partners.
It took me a while to figure out what you meant by distantly challenged, so, thank you, I appreciate it. So, I was talking about a journal club, so I'm imagining, oh, there would be a Skype component to the journal club, is kind of what I was imagining, but I've not tried that before, so it would be a new experience for me also, but one to which I'd be open.
I think email is a great way to find out more about what we do in our research lab. But also Skype, and the other thing you can think about doing as you're doing your research projects or wherever you're located is thinking about undergraduate research experience opportunities. So for example, I know that NSF has an undergraduate research experience where you can go to the school that you're interested in and working with someone, and you can apply to that program. And that's one way to get your feet wet into, the research experience itself, in addition to just learning about what research we do and what's out there in the field as well.
Thank you, Marilyn.
Maybe I'll just add one more thing to what was already said. Similar to NSF, NIH also has a similar mechanism and program around that too. So just some more food for thought.
Hi, my name is Sarah Florig. So as an undergraduate who's really interested in pursuing a PhD and applying for some of the graduate research fellowship programs that are available, what do you foresee in, I guess, building my own, from contribution to your lab, but my publications possible? Do you see possible publications coming from your lab and significant research projects that can be taken to symposiums and presented on? Are you comfortable with undergrads doing that, working with grant writing, things, just advancing my skills, but also in a way that contributes to your lab?
So, in being that I have this huge amount of undergraduates in my lab, part of being in my lab means that you not only get to do the research that I'm interested in, but I'm hoping that you'd be able to come up with ideas of your own, so I totally support that. In my group meetings, we do both research and literature group meetings where you get to present your research, but also you get to present your data. The reason behind that is so that we can prepare you to go to national conferences. So there's opportunities in my lab to support you to go to a meeting with me, and I love doing that because that helps you build your own network. To meet people that you might be interested in working with in the future. There's lots of opportunities locally here to do poster presentations, so we prepare you to do that. In terms of writing, since you're the one doing the research, then you should be the ones on the publication, at least from my perspective. So I encourage and I support writing initiatives in my group. So I teach you how to write a manuscript because I'd like you to be able to have your name as a first author in a publication, because that's really what's going to help you get into graduate school. In terms of graduate research fellowships, when you're thinking about NIH or NSF, it's also something I support. So I think the earlier you can have experience writing grants, or at least looking how we write grants as well, helping, that will help you in the long run.
I think probably her answer is true for all of us. The other big component of that, though, is you're all adults now and you're responsible for what you do. And we might be saying this is sort of the direction that you should be going, or this is something that you should be working on, or this is a skill set you need. But if you don't do it, then nobody is going to do it for you at this point. So really, what I have seen with undergraduate students in my lab is that there's a pretty wide diversity in terms of where they want to go and where they do go. And the ones that figure out that there are opportunities for them if they pursue it can go a long ways in terms of writing grants, writing papers, and presenting at national meetings. So.
Some people were still wiggling, and I mistook that for a question.
Hi there, I'm Rudy, and this is a question directed to Dr. Betty Izumi. Go BC.
The ice is, I can't hear. The ice is so loud.
Oh, sorry. This is a question directed to Dr. Betty Izumi. What's your approach towards nutrition and diseases?
What's my approach? Well, I'll preface by saying that this weekend I ate a lot of potato chips and pork ribs. But my approach to nutrition is really not focused on nutrients. I really try to promote whole foods, and I believe that the simplest nutrition advice for having good health is just to eat as few highly processed foods as possible. So in all of my projects, I'm thinking about how we can support local agriculture and work with and eat whole foods.
Thank you.
Hi, I'm Yvonne Jordan, and as an undergraduate looking at trying to gain research experience, you all talk about having undergraduates, but how, from the undergraduate perspective, do we apply? Find these labs?
We're gonna call in Dawn.
I mean, I do. They don't. But you know, the best person to answer that question is Marguerite. And not to delay the answer, but on Thursday we're having an entire session about how everyone in this room, all of you scholars, will become trained and ready to be placed in these very labs. So in the interest of wanting to make sure you have time to ask your questions about their content, I will defer that, Yvonne, we can talk about it, but I want to guarantee all of you that is a huge component of what you'll be learning this week. There's a very, I think, very detailed and supportive process and pathway in place to ensure that you're placed where you need to be to get the skills and knowledge that you need. And this is merely a sampling of some of the awesome opportunities that are in front of you.
Thank you, Dawn. Devin.
Hi, my name is Devin Slipkowski. I'm from Shemeketa. I actually have a question in regards to what Betty said at the end of her speech. Betty, you said that research things change all the time, and I kind of had visions of federal agents coming in and shutting down labs, but then I also had visions of new discoveries coming out. So maybe could you guys explain a little bit more or talk on the points of how has your research changed in your labs during your studies?
So let's see. 15 years ago, I started by studying how arterial pressure load causes fetal cardiac myocyte proliferation. And I did some different projects looking at different in vivo stimuli changing proliferation and maturation. And so that took about 7 years, and it was about 5 different kinds of stimulus, looking at different things like that the babies encounter, like low oxygen in utero, for example. And then I started wondering, you know, actually most people with heart disease have bad coronary arteries, and they have like ischemia or heart attacks. So then I thought, well, what's going on with the coronary arteries in these hearts? And I started actually looking at how the cardiac myocytes communicate with the coronary arteries. And then I realized, oh, if I'm looking at that, I actually should be looking at the fibroblasts too, because, you know, when you have a heart attack and you end up with a scar, it's the fibroblasts that are laying down the scar matrix. And so then I started looking at the fibroblasts too. So there's always an evolution in terms of, it's not like, oh, you know what, I'm going to study the liver. It's, you know, what's the next interesting question? And one thing that's really stuck with me is when I was a graduate student, one of my mentors said to me, you know, I'm old and I've thought all of the new creative thoughts I'm ever going to think. Now it's up to you. Like all of the really naive, crazy ideas you come in to ask me, like, oh, that's crazy. But you know what? It might be true. So like those ideas, those are the ones that we need to pursue. So I remember that, and I think, you know, when undergraduate students come and say, you know, oh, maybe it's the glucose. I'm like, well, that's kind of crazy, but you know, let's take a look at that. So that's sort of how the evolution happens.
Any other panelists want to respond?
Sure.
To that? It's a
I'll
great
speak a little bit to that. So I'm the social psychologist. I look at social psych aspects of health situations. I would say that one way that my work changes over time is kind of different kinds of methods that I use depending on what question I'm trying to answer at the time. So, you know, I might be doing a survey project. I've done some community-based projects where well, that's not a method, but it's a style of working with, you know, in collaboration with communities. Other projects I have might be using data that somebody else already collected. I might be using a qualitative method. And these are things that kind of depend on what questions I want to be asking at the time. There's also a cycle of, like, sometimes being out there collecting data, other times being kind of closer to the computer in writing or using data sets that someone else might have collected. So these are ways that there's kind of, I wouldn't say that there's exactly a set sequence, but there's more like a phasic kind of thing that happens that way. Anybody else?
So when I first started my independent research program, I thought I was going to develop catalysts to produce hydrogen and save the hydrogen economy. Right now, I do nothing with hydrogen whatsoever at all. That's what I wanted to do. So I wanted to develop these catalysts. And we started doing that a little bit, but we also had a small biomedical project in my lab. And now that has taken over everything. So nobody's working on the hydrogen. I'm pretty much going to like give up on even thinking about doing that, because I think what we're doing in my lab is taking off. So the students also drive where our research projects are going. So as you're starting off doing something, you're going to discover something new, and that's going to have a new question that you want to answer. And so you always find yourself shifting with new questions that you're trying to answer. The other way that my projects have kind of diverted from where I originally thought or they should go is from the collaborations that I'm forming. So by partnering with OHSU, I'm seeing within the last year a huge shift. I'm still doing a lot of the fundamentals, but they're asking me, hey, we need to look at this problem that we can't. Can you build me something to look at this problem? And so that got the project with the KCI Institute started. They approached me and said, you know what, this is a cool thing. I remember getting my eyes checked and talking about macular degeneration. And in the back of my head, I said, one of these days, I'm going to look at it, you know, I'll keep it on the back burner, and then I have a group that comes and asks me about it. So it's kind of interesting. By developing new partnerships and asking questions, things can really change. In terms of shutting your lab down, well, don't do anything wrong where you, go against the FDA or NIH or OSHA. That's the way that happens.
Wonderful. Two or three from one table here. Okay.
My name's Madison, and I just had a question for you, actually. I don't know if I'm going to butcher your last name, but...
Don't worry about it. Everybody just calls me Dr. Mack.
Okay, Dr. Mack. Specifically, since I noticed you're working on Alzheimer's, cancer, and macular degeneration, if we're more interested in one or two of those particular things, do we have the option to choose which direction we would like to go?
Yes. One of the things I encourage any student that wants to pursue research is find something that you're interested in working on. Because I think if you're not interested in working on that, then you're not going to have the passion that's going to drive what you want to do. So I always, present the different research projects that we have available and tell you where we need help. But ultimately, I give you that option on which ones you want to pursue. Because maybe you don't care about macular degeneration. Maybe you know somebody with Alzheimer's disease and that's where you want to go. Or maybe somebody in your family has cancer. So you might want to do a cancer-related projects. So I would say pursue the ones that you are passionate about and advocate for working on that project.
Thank you. My question is also for Dr. Mack and partially Dr. Jonker. I noticed on your slide that you work with the Oregon Primate Center, so I was just wondering if you could expand on that. And then I also wasn't sure how exactly you work with the sheep fetal cardiac myocytes.
Okay, so since I have the microphone, I'll go first. So a year ago, the Oregon Primate Center and the KCI Institute approached me about building imaging agents. My lab is a synthetic lab, so we build and design new chemistry to do things that people want or things that we want. We're now beginning to learn how to work with cells, so growing them, incubating them, which is something different than a synthetic chemistry lab would do. But I think it's something that we can handle very well. In terms of working with animals, I don't have the time to do that. And they're cute and everything, so I don't want to go work on rodents or macaque monkeys at all. And I don't have the expertise. So the Oregon Primate Center, they're equipped to handle all of that type of work. So whatever we build, they test in those systems. So they are trained, and I'm confident in their capabilities. So I just have to, basically, I make my materials, run over there in my car and drop it off. It looks really suspicious that I just go around in a circle, here you go, and then I leave. So I don't get to do any of the animal studies. But if a student of mine that was interested in learning more, that's one of the nice things about partnering. I can bridge that student with that lab. And if they wanted to do animal studies, they're happy to take on any of my students.
So our primary animal model is pregnant sheep. And sheep are seasonal breeders, and we do all of our in vivo studies from October through April. So, so graduate students in the lab that are full-time in the lab can do studies in the pregnant sheep. Undergraduate students typically have a lot of classes during those times. So we're happy to have students come watch and participate, but typically students can't participate at a high enough level to conduct those studies themselves. That's why we have the option of doing like PCR or Westerns from tissues from in vivo studies, or if they want to work with living tissues we have primary cell culture from those hearts. Yeah.
Thank you. Megan, last question. Take us home here.
My name is Megan. I know as a female in science and just a scientist that sometimes it can be really difficult, like you're just struggling with one thing and you're just like, oh my gosh, I just want to give up. This is really hard. Have you ever been in that situation And how did you deal with that situation where you just kind of wanted to give up and just like cry for like 5 hours?
No doubt we could have a whole session on that, so I'm just going to ask, in the interest of time, maybe one person respond, but very, very rich theme.
Okay, so I think the analog for myself would be when I was working on my dissertation, I had this sense I was going to die before this thing finished. You know, it was awful. It was just this terrible, it was like this cloud that would come in while I'm trying to write. So, I know this is going to sound made up, but this is what I did. I stopped, I set my dissertation aside, I wrote my will, I taped it to the file cabinet, and I went back to work. That's my advice.
What a cheerful way to bring us into lunch. Yes, okay, all right. We'll do that at the break. Will writing, okay. Well, scholars and guests, please join me in an enthusiastic thank you to our panelists. What a bounty.